Hello everybody, I'm going to start lecture 3, the carbohydrate metabolism, then we are going to follow the glycolysis. So let's talk on the content outline first. I'm going to give brief information on the carbohydrates, then we are going to discuss glycolysis. Afterwards, we are going to follow the pathways by the chart illustration, and then we are going to talk on the linker pathways on brief, and we are going to discuss the regulation since we are going to discuss the hormones in this step. First of all, where do carbohydrates meet our body? So we got some carbohydrates in order to gain energy, and they are going to enter into the body in the complex forms such as the disaccharides and the polymers. And for example, the starch is going to be composed of amylose and amylopectin, and then we are going to see the glycogen is another carbohydrate that we can find in animal cells. Starch is present in the plant cells. And if we discuss the polymer cellulose, it is also consumed, but it is not digested. And the first step in the metabolism of digestible carbohydrate is the conversion of the higher polymers to the simple and soluble forms, and they are going to be transported across the intestinal wall and delivered to the tissue. So we are going to see that this delivery is going to be the main source of energy. We have got big polymers, they are going to be degraded into small ones. That's the major aim in carbohydrate metabolism. So the breakdown of polymeric sugars just start in the mouth, and saliva helps to this process, which has an acidic pH of 6.8, and it contains a lingual amylase that starts the carbohydrate digestion because we couldn't digest the carbohydrates in the stomach. Stomach is reserved only for the digestion of the lipids and as well as the proteins. The action of the amylase, however, the enzyme that degrades carbohydrates, is just limited between the mouth and the esophagus. It's just a restricted area. Uh, Acid hydrolysis just contributes to the carbohydrate degradation. It is helpful in this case, but we have to think that this would be an important concept that we have to discuss in this case. And we need some specific gastric proteins and lipids only for the proteins and the fats respectively. Okay, they do not help carbohydrate digestion. Only the acid helps a little bit. So the mixture of the gastric secretions, the saliva and food, which is known collectively as chyme, can help the carbohydrate mixture move to the intestine, and the resultant glucose and other simple carbohydrates are transported across to the intestinal wall to the hepatic portal vein in general. And we can see then the carbohydrate mixture is going to be transferred to the liver parenchymal cells and other tissues. Therefore, they are going to be converted into fatty acids, amino acids, and glycogen. So there is a strict interaction between the carbohydrate metabolism and as well as the glycolytic metabolism in general. So if we discuss the basis of the structure of the carbohydrates, mainly we can find starch in plants and glycogen in animals, and they are composed of only one single unit glucose. So how can we degrade glucose is our question. Let's look into the answer of the question. The answer is glycolysis. Basically, it's very important pathway. So this is the sequence of reactions that converts glucose into pyruvate with the concomitant production of a relatively small amount of ATP. Our aim is to obtain energy here. So the glycolysis can be carried out anaerobically in the absence of oxygen, or an especially important pathway for the organisms that can ferment the sugars mainly. Also, we can find glycolysis in some other organisms, for example, yeast. This is the pathway utilized by yeast to produce the alcohol present in beer, and it serves as a source of raw materials for the synthesis of our other compounds. For example, 3-phosphoglycerate can be converted into serine, while the pyruvate can be aerobically degraded by the Krebs or TCA cycle, or the 3-carboxylic acid cycle, in order to produce much larger amounts of ATP. Only the difference is that in glycolysis we can form only 2 ATP as a net product, but as a if you compare the Krebs cycle, we have to obtain less amount of ATP when glycolysis is considered. So glycolysis only tend to obtain 2 ATPs, but if you just imagine Krebs cycle, we have to obtain more ATPs from this Krebs cycle or TCA cycle, or in other words, the citric acid cycle. So the oxidation of glucose is just maintained either to lactate, or even we can see that or the pyruvate. And under anaerobic conditions, the dominant product in most tissues is just the pyruvate, and as well as the pathway is just known as the aerobic glycolysis. So when oxygen is going to be depleted, as for instance during the prolonged vigorous exercise, the dominant glycolytic product in many tissues is just said to be the lactate. That's very important. And the process is called anaerobic glycolysis. So we got aerobic glycolysis and anaerobic glycolysis. We have to be very careful in imagining this case. So what we're going to derive as an energy from the glucose oxidation, that's another question. So the aerobic glycolysis of glucose into pyruvate just requires 2 equivalents of ATP in order to activate the process, just with the subsequent production of 4 equivalents of ATP and 2 equivalents of nicotinamide dinucleotide reduced form is obtained. And the energy which is derived from the glucose oxidation is just seen in the slide, that we can see that the conversion of 1 mole of glucose into 2 moles of pyruvate is just managed by the net production of just 2 moles of each of ATP and nicotinamide dinucleotide reduced form. So we start with glucose plus 2 ADP and as well as 2 nicotinamide dinucleotide oxidized form, and as well as we add 2 inorganic phosphates. After this, we can obtain 2 pyruvates, 2 adenosine triphosphates, 2 nicotinamide dinucleotide reduced form, and 2 hydrogens at the end of this reaction step. So the nicotinamide dinucleotide generated during the glycolysis is just used to fuel the mitochondrial ATP synthesis via the oxidative phosphorylation. We can produce either 2 or 3 equivalents of ATP depending upon whether the glycerol phosphate shuttle or even the malate aspartate shuttle is just used to transport the electrons from the cytoplasmic nicotinamide dinucleotide into the mitochondria. So we have to obtain some electrons, and these electrons are going to be just transported from the cytoplasmic nicotinamide dinucleotide into the mitochondria as well. So we got some kind of a net yield for the from the oxidation of 1 mole of glucose into 2 moles of pyruvate, either 6 moles of or 6 or 8 moles of ATP. And in the complete oxidation of the 2 moles of pyruvate through the Krebs cycle, it would yield an additional 30 moles of adenosine triphosphate previously, and the total yield is just either 36 or 38 moles of ATP from the complete oxidation of 1 mole of glucose into carbon dioxide and water previously, and the current case is just said to be around 30 to 32 ATPs. We can see that there are 2 different approaches. One of them is the previous approach that we already know from our high school days. We obtain 38 or 36 or 38 ATPs, but in the new approach, which is valid from 2002, we have to obtain between 30 to 32 ATPs. This is a new one, but still the second one, the new approach, is under debate. It is not definite, okay? So the pathway of glycolysis can be seen as consisting of 2 separate phases. The first one is the chemical priming phase requiring the energy in the form of ATP, and the second is just considered as the energy yielding phase. So let's have a break.